Okay, good morning. I hope that everybody's maybe slept a little bit, is going to get some perks out of virtual school day. This is going to be a screencast lesson. I'm going to post this video once it's done to the drive, and you can watch it in class to kind of recreate the lesson the way we normally do in class. What I'm going to do is I'm going to tell you when to pause the video and then either answer the questions or let's look up online or do the kind of little engagement and discussion activities we do um, in class time. If you're with somebody else in class, then please actually discuss with them. If they're not, then just try to think through the questions yourself, but really give yourself a minute and try and think through this. Don't just skip right through the video. You'll get a much more effective lesson, and we're not going to go over this material again. This counts as your lesson. We will, of course, have a review day, but you need to really be on it. So lesson number 2 is the ultrastructure of cells. First things first, you need to do your Edmodo quiz. It's already up on Edmodo, so pause the video and go to your quiz. Okay, hopefully you have done your Edmodo quiz now. I will be checking to see if they're done as part of your attendance for today's class. And then take a minute and kind of check in with yourself how practical 1 is going. I know most of you still need a little bit of time to finish up your drawing. We would have done that in class today, but of course we can't do that from home. So I'll try and find some time for you on Thursday to do it. But you can always come at lunch, after school, before school, during your study block, any of those times to work on it as well. Okay, so subtopic 1.2, here's what we need to learn today. Prokaryotes have a simple cell structure without compartmentalization. Eukaryotes have a compartmentalized cell structure. Electron microscopes have a much higher resolution than light microscopes. And applications, student structure and function of organelles within exocrine gland cells of the pancreas and within palisade mesophyll cells of the leaf. Prokaryotes divide by binary fission. Drawing of the ultrastructure of prokaryotic cells based on electron micrographs. Drawing of the ultrastructure of eukaryotic cells based on electron micrographs. And interpretation of electron micrographs to identify organelles and deduce the function of specialized cells. So first things first, what is a prokaryotic cell? So you should have learned this in middle school, earlier in high school. You should have heard it multiple times already. So take a minute and answer these 2 questions. So what key feature of prokaryotic cells differentiates them from eukaryotic cells? And what kind of organisms are prokaryotes? So pause it and answer the questions. Okay, so the key feature I was looking for in the first question was about the nucleus. It's the fact that eukaryotes have a true nucleus. The prefix EU means true and karyote is nucleus. And prokaryotes have an early nucleus, so it doesn't actually have a nuclear membrane. If you look at the picture, it's just kind of a clump of DNA. It's called the nucleoid in a prokaryotic cell. And then what kind of organisms are prokaryotes? So remember topic 5 was evolution and diversity. Examples of prokaryotes include bacteria, archaeans. Those are 2 main groups of prokaryotes. What the IB says they want you to be able to draw of a eukaryote should include the cell wall, pili, those little kind of hairs coming out of the prokaryote, and the flagella, as well as a plasma membrane, including cytoplasm, that contains 70S ribosomes and a nucleoid with naked DNA. We'll go through what a 70S ribosome means a little bit later in today's lesson, but those are the things you need to be able to draw. So take a minute and practice drawing the prokaryote with all those different features labeled properly. Remember, label lines don't cross. Okay, so I would like to see your drawings next class. You can send me a picture of them later on today as well through email or through Edmodo, and then I'll take a look at them as well. Okay, so binary fission is the process by which prokaryotes replicate, actually reproduce. And if you notice here, we have the diagram showing binary fission of bacterium, because a lot of prokaryotes are bacterium. What kind of reproduction is this? This is asexual reproduction, right? They are not reproducing sexually. They have this no sexual reproduction happening here, only one bacterium dividing by itself. So it's asexual reproduction. Notice the positioning of the DNA while the division is happening. Why do you think that is the case? So if you notice, the DNA is kind of being sent and copied towards each extreme, the left side and the right side of the bacterium as it elongates itself, and then it separates itself. And this is the way that bacteria will make sure that each product bacteria has a complete copy of the DNA. In eukaryotes with much more chromosomes, we do the whole mitosis process. Meiosis is much more complicated, but in prokaryotes, they only have one chromosome and a plasmid, so they don't need to have that whole orchestrated process of metaphase and anaphase and organizing all the DNA. And the last question, what kind of evolutionary advantage does this give the prokaryotes? So pause the video and think about this one. So the big thing about asexual reproduction is it's not good for diversity, but it's great for speed. You can reproduce a lot of offspring really quickly. So as long as you're well suited to your environment, you can really have a giant population growth very quickly. It's a major advantage of binary fission.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
There's the speed, really quick reproduction. Okay, so that's prokaryotic cells. Now, eukaryotic cells are more complex and they are compartmentalized. They have compartmentalization. That's the word the IB wants you to use. So take a minute and look at these two diagrams of eukaryotic cells and discuss with yourself, with your partner, with your cat, with whoever, how does compartmentalization allow eukaryotic cells to be more complex? Okay, so what you should be noticing here is we have different organelles. We have mitochondria and chloroplast and a nucleus and endoplasmic reticulum and the Golgi, lysosomes, all these different things that allow the cell to carry out different processes inside those organelles. And the next thing, why we need to have these being compartmentalized within their own nucleus, within their own membranes, is to allow them to have the enzymes they need to complete their task without affecting other parts of the cell, to allow them to kind of organize the various materials they need to complete their task and make things much more efficient. So if you think about the prokaryotic cells, they are much simpler structure. They don't have all these compartmentalized organelles. They just have a clump of DNA and a couple things, and that's it. Right? Some ribosomes, some cytoplasm, none of these complicated organelles to help them. Next question, what kind of organisms are eukaryotes? So connection again to topic 5, pause the video, take a minute and think about it. So eukaryotes are all the other kingdoms of life, everything in the domain Eukaryota, if you remember your domains and kingdoms Protista, kingdom Fungi, kingdom Animalia, and kingdom Plantae. So plants, fungi, animals, and prokaryotes are all, not prokaryotes, protists are all eukaryotes. What the IB would like you to be able to do when you're drawing a eukaryotic cell is show the plasma membrane enclosing a cytoplasm that contains 80 S ribosomes. Remember, we're going to differentiate between the two kinds of ribosomes in a little bit, and a nucleus. Mitochondria and other membrane-bound organelles are present in the cytoplasm. So you could include the Golgi body, lysosomes, endoplasmic reticulum, ribosomes, all those kinds of things. Some eukaryotic cells have a cell wall, and some are mostly plant cells. So take a minute, pause the video, draw the cell, and like the prokaryotic cell, I want to see a picture of this drawing sent to me sometime today. Okay, through Edmodo message, through email, something like that is fine. Okay, now comparing animal cells and plant cells. On the left, we have an animal cell. On the right, we have a plant cell. So I'm going to give you a minute to really think about this. And you've done this multiple times, high school, middle school, you should have done this. Answer these two questions. What do plant and animal cells have in common and why? And what are the differences? Why? Really think about how those differences and similarities allow them to do their function, to survive, to thrive, to do what they have to do as an animal or a plant, or the organism containing those cells. Okay, pause the video and take a think. Okay, so hopefully you notice that both have a nucleus with a nucleolus. They are eukaryotes, they have to have a true nucleus. They both have an endoplasmic reticulum, smooth and rough, that allows for transcription and translation to happen to make proteins from the DNA. They both have a Golgi complex, Golgi body, which allows them to process proteins and move around the cell. And they both have mitochondria, which help them to get energy, cellular respiration. They both have ribosomes, different sizes, but the same kind of idea of ribosome that allows them, so the same size for all eukaryotes here, to translate the RNA into a protein. The big difference you should be noticing is that we have chloroplasts and a cell wall in plant cells where we don't have those in animal cells. So plant cells, of course, have to do photosynthesis, and the cell wall helps them have that supported structure they need to be able to remain turgid, to remain kind of stiff in shape, whereas animal cells instead have kind of a cytoskeleton along the inside of the plasma membrane. Okay, so you need to know the structure and function of several different organelles. Some are only found in prokaryotes, some are found in plant cells, and some are in animal cells. BioNinja, as we know, is a great resource. I'm not going to go through every organelle with you because you've heard them a million times. It's not a good use of our time, so what I'm going to ask you guys to do is to go into BioNinja and look up all the different organelles. There's 13 in total. If we were in class together, I would have each of you do 3, maybe I would do 1, and then we'd have all 13 done to kind of share with each other. The big thing here when you're reading this and adding to your notes is talk about how structure relates to function. So how does the structure of a chloroplast allow it to do its function of photosynthesis? How does the structure of a membrane allow it to do its function of letting things to come into and out of the cell? So pause the video and take a good 10-15 minutes to do this. Okay, hopefully that was good and you got some good information in your notes about the different organelles. Next, we're going to differentiate between the two types of ribosomes. So remember earlier we talked about prokaryotes have a 70S ribosome and eukaryotes have an 80S ribosome. So the S is just a unit of measure they use for ribosomes. So you look, we have a slightly bigger ribosome in the eukaryote, which makes sense, it's a bigger cell. So compare and contrast the more detail of these ribosomes and say how are they the same, how are they different, and pause the video to do so. So hopefully what you're noticing is that they are pretty much exactly the same in structure. The difference you're noticing here is the size, is that prokaryotes are smaller, they have a 50S subunit and a 30S subunit, so they have an overlap of 10S. So the total size from top to bottom of the ribosome is 70S. And the eukaryote, they have a total size of 80, the top subunit being 60S, and the bottom being 40S. So it's an overlap of 20S in this case. They both have the RNA. It's going to go through them. They're, again, slightly bigger in the eukaryote than prokaryote. But it's all about the size. Okay, so as a big part of topic 1, we talk about light microscopes, practical when you're using the light microscope and how to look at it. We already discussed kind of cell theory and how that came to be using microscopes and how it's changed the way that we view the world. Without knowing without the microscope, we could never see things that are microscopic, which seems like a very obvious statement now, but in the 1600s, people had no idea there were things they couldn't see with the naked eye. So electron microscope allows us to see a really high level of magnification that makes viruses visible and other really, really small things we could never see otherwise. So So take a careful look at this diagram and notice the higher magnification you can see things. So the naked eye, you can see a whole person, a child, a hand, a finger, just fine. Light microscope, you get into looking at hair, blood cells, bacteria. Electron microscope, overlap there a little bit with blood cells, bacteria, viruses, DNA, glucose, and atoms, even electron microscope. Electron microscopes are big, they take up like a small room, and they are very, very expensive. I would love to use one someday. So here is the same image of the same specimen under a light microscope and under an electron microscope. This is a radiolarian. It is a protist. You'll notice the electron microscope does allow us to examine things that are much smaller. There are some disadvantages to the way it looks. First thing, electron microscopes are crazy, stupid expensive. $50,000 to $120,000 for just the microscope. It needs a special room with very careful air conditioning and kind of cleanliness maintained, and requires a lot of electricity to actually use. It takes a long time to prepare a specimen to view it under an electron microscope. You have to prepare it in a very specific way with certain chemicals and be highly trained to do so. And there's no color in a microscope image, electron microscope image. So if you look at the first one here, it's two-dimensional. It's not as detailed, but it's way cheaper, and you can see color if there was color present in this image, but there's not in this particular one. Whereas in the electron microscope, it's way clearer, much, much more detail, but you have to really make your choices about which things you're going to do and see. Okay, so one of the skills we need to work on for today is examining electron micrographs. So these images taken with an electron microscope are actually challenging to read, and you have to learn how to do so. So we're going to do a bit of practice today doing that. So first thing, this image is a prokaryotic cell or eukaryotic cell, and why? Hopefully you said it's prokaryote, and the big reason why is there is no nucleus. A clump of DNA in the center called the nucleoid, and that's it. There's no membrane-bound nucleus. It has to be prokaryote. Identify the cell wall, plasma membrane, cytoplasm, ribosomes, nucleoid, and pili. So pause it and try and see those things. I'm going to exit so I can actually point things out to you. So we have here the cell wall is a thick outer structure. The plasma membrane is just inside the cell wall. Then you have a cytoplasm as all this kind of goop that's all filling the cell. The ribosomes are these little dark circular dots. Those are the ribosomes. The nucleoid is the central piece of DNA, and the pili are these little kind of projection hairs on the outside. Okay. So we're going to do a few of these to practice together. Okay, here another cell. Is this a prokaryote or a eukaryote, and what do you know about it and why? Well, this is definitely a eukaryote because the nucleus has a membrane around it, and it is a plant cell because I have chloroplasts here. So identify the cell wall, cytoplasm, nucleus, nucleolus, vacuole, chloroplast, and mitochondria. So first thing, cytoplasm is any of this space that's not filled with organelles. It's all cytoplasm. The nucleus is here. It's the membrane nucleus. And then inside it, I have the nucleolus. The vacuole is here. Plant cells have one big vacuole for structure. The chloroplasts are here. You can see the kind of the stacks of grana and stoma here that are going to allow for photosynthesis. And the mitochondria are here. You can tell because they have this internal membrane structure, the cystae, that allows it to do respiration very effectively across all those different membranes. So you should be practicing getting better at doing this. Hopefully with practice, you'll start to see things better. Okay, so with this cell, take a minute, pause the video, and try and figure out what kind of cell it is and identify all those structures. Okay, so this is again a eukaryotic cell. You can tell because we have this big old nucleus over here. It has plasma membrane here on the outside. That's the plasma membrane, quite thin, this can pass in and out. The nucleus, of course, the big structure. The cytoplasm is anywhere in here with no structures. Ribosomes, all the little black dots are ribosomes on the outside of the nucleus. The endoplasmic reticulum, all this folding kind of lines around the nucleus, that's all the ER. Okay, and you can see it's got the ribosomes in it, so that's the rough ER. The Golgi apparatus is this kind of circular structure up here. It's kind of curved looking. Lysosomes are these little circles coming off the Golgi, and mitochondria, here's the stable mitochondria. You can see the kind of internal cystae structure. Okay, we've got another one here. So looking at the actual pancreas, it's asking you to talk about the pancreas and its structures. So take a minute and examine the pancreas image here. So remember pancreas from topic 6. Here's a diagram of it, and here's an actual cell electron micrograph image. So take a minute and pause and take a look at that.